Inside every living organism, trillions of cells face a critical decision every single day: when to divide and when to wait. Get this timing wrong and the consequences can be catastrophic, leading to developmental defects, tissue damage, or cancer. For decades, scientists understood that cells divided, but the hidden machinery controlling this process remained a mystery. One researcher would change that forever. The cell cycle is the carefully choreographed sequence of events that allows a single cell to duplicate its genetic material and split into two daughter cells. This process unfolds in distinct phases: a growth period, DNA replication, another growth phase, and finally division itself. Each step must occur in perfect order, with flawless precision. But what ensures this precision? What prevents a cell from dividing before it's ready, or stops a damaged cell from passing on corrupted information to the next generation? In the 1960s and 1970s, Leland Hartwell, working at the University of Washington, approached this question with an elegant strategy. Rather than trying to observe the cell cycle directly in complex human cells, he turned to a simpler organism: Baker's yeast. These single-celled fungi divide rapidly and are easy to manipulate genetically, making them ideal subjects for uncovering fundamental biological principles. Hartwell's breakthrough came from a deceptively simple idea. If specific genes controlled the cell cycle, then mutating those genes should disrupt the process in predictable ways. He systematically created yeast mutants and observed what happened when they tried to divide. Some mutants got stuck at specific points in the cycle, unable to progress further. These arrest points revealed something profound. The cell cycle wasn't just a continuous flow of events, but rather a series of regulated transitions, each governed by specific genetic controls. Through painstaking work, Hartwell identified more than 100 genes involved in cell division control, which he called CDC genes, short for cell division cycle genes. Each gene played a specific role, acting like a molecular switch that had to be flipped at precisely the right moment. But perhaps his most significant discovery was the concept of checkpoints, critical control points where the cell pauses to verify that everything is ready before proceeding to the next phase. These checkpoints function like quality control inspectors on an assembly line. Before a cell replicates its DNA, checkpoint proteins verify that conditions are favorable and that the cell has grown large enough. Before division begins, another checkpoint ensures that all DNA has been copied correctly and that any damage has been repaired. If problems are detected, the checkpoint halts the cycle, giving the cell time to fix the issue, or if the damage is too severe, triggering self-destruction to protect the organism. The implications of Hartwell's work extended far beyond yeast cells. Researchers soon discovered that the same fundamental control mechanisms exist in human cells, preserved across hundreds of millions of years of evolution. The CDC genes Hartwell identified in yeast have direct counterparts in human biology, controlling our own cell division with the same molecular logic. This discovery revolutionized our understanding of cancer. Cancer is fundamentally a disease of uncontrolled cell division, where cells ignore the normal stop signals and multiply without restraint. Hartwell's work revealed that many cancers arise when checkpoint genes become mutated or disabled. Without functioning checkpoints, damaged cells divide anyway, accumulating more mutations with each generation and eventually forming tumors. Understanding these control mechanisms opened new avenues for cancer treatment, allowing researchers to develop therapies that target the specific molecular switches governing cell division. In 2001, Hartwell shared the Nobel Prize in Physiology or Medicine for his groundbreaking discoveries. His work demonstrated that the most complex biological processes are governed by identifiable molecular mechanisms that can be studied, understood, and ultimately manipulated for therapeutic benefit. The hidden controls of the cell cycle, once invisible to science, now stand revealed as one of life's most elegant regulatory systems. From a single yeast cell to the trillions of cells in the human body, the same ancient checkpoints stand guard, ensuring that division occurs only when the time is right. Hartwell's legacy lives on in every cancer treatment informed by cell cycle biology, and in our continuing efforts to understand and harness the fundamental mechanisms that govern life itself.